Welcome to the science video. Are you fan of laboratory experiments? If so, you're lucky because in this video, we're going to present the common laboratory apparatus as well as the do's and don'ts when inside the laboratory.
Here are some of the laboratory safety rules a student must follow. Number 1. Follow the instructions. If you're unclear about any point or have questions, get them answered before starting. Number 2. Keep snacks out of the lab. Food and drinks should never be consumed in a lab. Number 3. Don't sniff the chemicals. Tasting or smelling some chemicals can be dangerous or even deadly, so it is prohibited. Number 4. Dispose of waste properly. Much of the waste created in a lab needs to be disposed of in something other than just the regular waste bin. Number 5. Identify safety equipment. If something goes wrong while you're in the lab, you need to know where the safety equipment is located so that you can start using it right away.
Number 6. Think safety first. It's impossible to eliminate all accidents from a lab, but you can take the right steps to prepare yourself for one. It could prevent a small problem from turning into a larger one.
Number 7. Dress for the lab. From the moment you walk into a lab, you need to be dressed properly from head to toe. This means wearing long pants, a lab coat, safety goggles, covered shoes, and any other protective gear required by the lab.
Number 8. Don't play the mad scientist. Another important safety rule is to act responsibly in the lab. Don't do randomly mixing chemicals to see what happens. Number 9. Leave experiments at the lab. It's important for your safety and safety of others to leave your experiment in the lab. Don't take it home with you. You could have a spill or lose a specimen or have an accident. Number 10. Don't experiment on yourself. You won't gain superpowers or discover the secret to eternal youth. More than likely, whatever you accomplish will be at great personal risk.
Today, I'm going to discuss or to introduce to you the basic apparatus used in laboratory. The first one is beaker, beer flask, wire brush or test tube brush, dropper, thermometer, mortar and pestle, alcohol lamp, petri dish, test tube holder or tongs, watch glass, wire gauze, and ring.
Eyepiece, it contains the magnifying lens. It is where we look through. Force adjustment, the large knob which moves the body tube to produce large changes in the focus. Body tube, it holds the eyepiece and lenses in position. Fine adjustment, the large knob which moves the body tube to produce small changes in focus. Revolving nose piece, it is a circular metallic piece holding the magnifying lenses to the tube. It revolves freely so as to select the required lenses needed to focus. Objective lens, it covers a wide field of view and they are useful for examining large specimens or surveying many smaller specimens. Arm, it supports the body tube. Stage, the flat surface on which the slides are placed. Stage clips, they hold the slides in place. Iris or diaphragm, it relates the amount of light entering the body tube. Base, it provides support for the microscope. Mirror or illuminator, provide even high-intensity light at the place the field aperture so that the light can travel through the condenser to the specimen.
Bacterial culture. Bacterial culture is a method that allows the multiplication of bacterial cells in a culture medium under controlled laboratory conditions. There are 5 basic steps in bacterial culture. First one is preparation of medium. In preparing culture medium for any microorganisms, the primary goal is to provide a balanced mixture of the required nutrients and concentrations that will permit good growth. Step 2. Adjustment of pH medium. For the growth of the particular microorganisms, the medium should have a specific pH. Step 3. Preparation of snaps and salons. All the materials like cotton buds and tissue should be sterilized to avoid contamination of the sample. Step 4. Pouring of plates. In this step, we need to pour the upper and finish. By this process, a larger surface area is created for the growth of microbes. Step 5. Inoculation of bacteria in nutrient plants and agar plates. Before streaking, you need to sterilize first the wire loop at the petri dish to avoid contamination. In streaking, form a zigzag lines to reduce gradually the number of cells along the line and also to get single colonies after incubation.
I hope the steps are clear. Now, let's watch a short clip on culturing bacteria.
Thank you everyone for staying with us throughout this video. We hope that you are inspired to be future scientists and contribute greatly in our society. On behalf of my co-future biotechnologists from BIS Bio2B, thank you very much and God bless on your journey.
Acknowledgement. We would like to thank the following: San Martin Agro-Industrial National High School, Mr. Peter Z. Manriquez, Laboratory in Charge, Mr. Raymart H. Boy Banting,